skeletal muscle, you have these sarcomeres that are bounded by the D lines, and in the middle we have these, of the sarcomere, we have these thick filaments with these myosin crossbridge heads. So what do they do when activated? They reach up and grab these active filaments and pull them toward the center of the sarcomere, and that's what causes muscle shortening. So, just think about it. Just put your fingers from the insertion origin of your sternocleidomastoideus, and you go, wow, that's quite a bit of shortening. Quite a bit of shortening. So those filaments slide over one another when those crossbridge heads are ratcheting to the inside. Now, there's more going on than just a thick filament, a thin filament lying next to one another. Thin filaments are pretty complicated, and there are these balls of globular actin wrapped around, generate two filaments, actin filaments, that are wrapped around each other, and there are these myosin crossbridge binding sites, very hard to see them, that are found on actin. And in the unactivated state, the protein tropomyosin is this long blue protein. This is lying over top these myosin binding sites. So this actin binding site right here can't get to that binding site when that tropomyosin is sitting there. Associated with both the actin filaments and the tropomyosin are these reddish colored troponin proteins. And troponin, it's a calcium binding protein. Calcium will bind right here, and that's going to cause a conformational change in troponin, which will then cause a conformational change. The troponin will pull the tropomyosin away from those myosin binding sites that are found on actin. So we were just getting through these. Calcium is really the trigger that gets things going for myosin crossbridge heads binding those binding sites on actin. And when we have low cytosolic calcium, there's no calcium associated with that troponin. Those tropomyosin proteins overlie those binding sites, so there's no binding and therefore no contraction. Remember, contraction is tension generation. But when there is sufficient calcium in the system, calcium binds troponin. We're going to see in calcium binds troponin in skeletal muscle, in cardiac muscle. Calcium binds calmodulin in smooth muscle. So there's a difference there. So here in skeletal muscle, calcium binds troponin. It causes a conformational shift, pulling that tropomyosin away from those binding sites, and therefore you can have engagement. Once the myosin crossbridge sites engage those binding sites, as soon as they bind, there's a couple of very quick actions, and then this head is going to ratchet toward the inside, pulling that thin filament to the center of the sarcomere. Okay, so calcium's a big player in getting that rolling. So we get calcium from, each one of these round tubes, those are myofibrils, and they are surrounded by a very elaborate sarcoplasmic reticulum. So here we have this system that there are a number of calcium binding proteins in here that attract calcium. And there's channels, so there's a lot of calcium in here, and there's pumps to constantly be pumping active transport pumps, pumping calcium into the sarcoplasmic reticulum. And there are channels in here that when those channels open, calcium can just flow out of the sarcoplasmic reticulum into this area, into these myofibrils, flowing down its chemical concentration gradient. And so you can have a nice uniform contraction of the whole apparatus. So up here, this is the sarcolemma, or the muscle fiber plasma membrane, and we were talking about this when we finished off So, okay, we have the release of acetylcholine. So we have action potentials coming down this A alpha motor neuron. So skeletal muscles are innervated by A alpha motor neurons and those A gamma motor neurons, but those are just for the intrafusal muscle fibers underneath. So here we have action potentials coming down, causing calcium to come inside the synaptic terminal, leading to a whole series of steps. Remember those synaptic steps that cause the release of the neurotransmitter. So acetylcholine is going to bind these nicotinic cholinergic acetylcholine receptors. They will open up, primarily sodium is going to come through. It's going to depolarize this patch membrane called, right here, called the motor end plate. And if it depolarizes it enough, that current's going to pass up here to the rest of the plasma membrane, and that's where we have voltage-gated sodium and potassium channels. So we get action potentials, and those action potentials will literally race across that membrane and go down these invaginations of the membrane called the T-tubules or transverse tubules. So these are literally invaginations of the plasma membrane. So action potentials can race along the surface here. Action potentials can go along here. So guess what? If you have action potentials here, what kind of channel proteins do you have here? Voltage-gated sodium and potassium channels here too. So we have this very elaborate network. So action potentials racing across here, coming down, and then it sets off a signal transduction signaling to the sarcoplasmic reticulum. So we get depolarization or action potentials coming down the T-tubules, and that activates what's called dihydropyridine receptor, which is right there, which then activates a ryanodine receptor, which the bottom line is when you have activation, so action potentials do not cross. I don't want you to think action potentials cross from the transverse tubules, the T-tubules, to the sarcoplasmic reticulum. They do not. But action potentials will activate these. There's actually three elements here. This dihydropyridine receptor, once it's activated by depolarization, it activates this ryanodine receptor, which in turn activates a calcium channel. It causes that calcium channel to pop open. Now, in the past, for years, I didn't really have students worry too much about what are the players there. Turns out that there are some diseases that impact these right here. So you do need to know this sequence. So action potentials coming down, going to activate the dihydropyridine receptor, which in turn activates this ryanodine, and it's also called the ryanodine positive receptor, which in turn will then open sarcoplasmic reticulum calcium channels. And there's so much calcium here that it'll just flow out by simple diffusion. It's going down its chemical concentration gradient. When calcium gets to a high enough level to bind a sufficient number of troponin subunits, that will cause the tropomyosin to shift, and that will cause thick and thin filament engagement. And as soon as it does that, as soon as they engage, you have that cross-bridge head ratcheting toward the middle. So there's quite a bit of stuff on this figure 926, and do read page 250. Oh, wait a second. Those pages are wrong. We'll change that to the revision. But there is a very short description of this in your book that I want you to just read, just so you know that you take action potentials here and you lead to signals that lead to opening of the calcium channel. Make it simpler for yourself in order to understand it. Now this is really quite involved, and I don't think you necessarily have to know. I want you to look this over. I want you to understand what's going on, but I'm not going to, for the first time in my life, I'm going to tell you this. I'm not going to have you just cold memorize this. I don't know if it's worth all of your effort. You'd probably end up forgetting it anyway, but there's this sequential step-by-step action that occurs during what's called cross-bridge cycling. This is cross-bridge cycling. So we have the release of calcium. Well, let's start in this slide. So we have tropomyosin overlying those binding sites. We have an energized myosin cross-bridge head, and I say it's energized, it has an ADP and an inorganic phosphate bound to it. This gives it several things. It's just like a gun cocked and ready to go off. This has incredibly high affinity for actin. It wants to bind actin. And it's also, like I say, it's a gun already cocked. It's loaded, it's ready to go. But it can't get to those actin binding sites, okay? So, we have the release of calcium from the sarcoplasmic reticulum. That's going to cause the tropomyosin to shift. We will have immediate engagement of that cross-bridge head, and as soon as it engages, what happens? That ADP and inorganic phosphate, that's their signal to kick off. And as soon as they kick off, that head ratchets inward. That's called the power stroke. So a lot of things going on there. And as soon as it has the power stroke, it immediately loses its affinity for actin, and it disengages. Well, excuse me. ATP is going to bind first, and then that's going to lead to disengagement. So ATP binds, that leads to rapid disengagement. This is really, really fast. And then what does it do? That ATP is hydrolyzed to ADP and inorganic phosphate. What does that do? That recocks the head. It gives it high actin affinity once again, and it's energized for the next power stroke. Okay? So this goes on over and over and over as long as calcium is elevated. As long as calcium is elevated, we have this type of cycling going on and on and on. Okay? So I want you to understand here how ATP is involved, how calcium is involved, how you can set up a situation where during one muscle contraction, this same cross-bridge head will bind, ratchet inward, release. Bind, ratchet inward, release. So it's kind of like it's just pulling on a rope. It's pulling on a rope and then regrasping, pulling on a rope, and just constantly bringing something closer to it. And you have all these cross-bridge heads doing this. So ATP is very much involved with powering this. Calcium is very much needed to initiate this. So just at least appreciate that. So I did want to mention, and I just briefly mentioned the other day, so the term contraction, muscle contraction, refers to tension generation due to cross-bridge cycling. So when you hear a muscle contraction, that means there's tension generation going on due to cross-bridge cycling. Now, oftentimes when we think of contractions, we think of this, shortening of a muscle, shortening of a muscle. So here we have a relaxed set of sarcomeres, and here there's a shortening contraction, That is called a concentric contraction. When you have tension generation due to cross-bridge cycling, and it leads to shortening of the sarcomeres, that is a concentric contraction. A concentric contraction is a shortening contraction. There's actually 3 types of, I only wrote 2 up here, but there's 3 types I want you to know about. Now, whenever you're, when you go to sit in the chair, you don't just plop down. You slowly lower yourself down. Guess what? You're contracting your quads, but your quads are lengthening as you are sitting down. When you go down the stairs out in front of the building, you're contracting your quads, you can't stand up. But you're actually, those muscles are lengthening. You're not causing such a muscle contraction to lead to shortening. You are just contracting the muscle enough to arrest, to slow down your movement. So your quads are contracting. When you're going up steps, it's a shortening or a concentric contraction. When you're going down, you're just slowing your movement, so there's still quad contraction, but the muscle's actually lengthening. That's an eccentric contraction. An eccentric contraction is when you have tension generation due to cross-bridge interaction, but the muscle is actually lengthening. So really what's going on during eccentric contraction, you're only contracting a limited number of myofibrils, not enough to shorten, but enough to at least slow things down during that movement. And then there's the isometric contraction. Iso means equal, metric means movement. This is where you're contracting, but there's no movement. That is contraction with no movement. So eccentric is extending or lengthening. Concentric is going to the center or shortening. But when you just hear the term contraction, all you know is there's tension generation due to cross-bridge cycling. That's all you really know. This doesn't really fit too many places, but so I'm just going to throw it in right here. There's so much in the book about twitches and tetanus and so forth, and we're not really going to talk about those things right here. But here is the action potential that's racing across the muscle circle lemma. And it only takes about 5 milliseconds for that entire action potential. Then there's a short period of time before you start seeing tension generation from that muscle contraction. So this is just to show you that action potential is very, very fast. Think of all the steps that have to happen. So you have that action potential up on the surface. It has to go down the T-tubule. It has to activate that dihydropyridine receptor, then the ryanodine receptor, then open the calcium channel. Calcium has to diffuse out. Calcium has to bind troponin. Tropomyosin shifts. And then you get cross-bridge interaction with the actin. You have the power stroke, and you start generating tension. So a lot of steps have to take place. So that's why there's this latent period. And it takes a while to get everything up and running to have your maximum tension generation. So just know that it's not instantaneous, but it's still pretty quick. So from the time you have the initiation of the action potential, it's only about 10 milliseconds before you start seeing tension generation. So it's a fast process, but it's not instantaneous, because think of all those steps that you have to go through, okay? And that's what I want you to do. Think of those steps, think of those steps, think of those steps. Now, here's just kind of in moving pictures a little bit about how this works. So here we have a motor end plate. So right here would be nicotinic acetylcholine receptors. So these are ligand-gated receptors here at the motor end plate. This is the motor end plate. This region between the synaptic terminals and the motor end plate, that's called the synaptic cleft. Here's T-tubules, here's the sarcolemma, sarcoplasmic reticulum, sarcomeres. So, one thing about skeletal muscle, there's a few organs in your body that, well, I shouldn't say they're dumb, but they don't do anything unless told to do so. Skeletal muscle and lung are kind of the same, but they're just hanging out until they're told what to do and when to do it precisely. Skeletal muscle contraction requires neural stimulation. We're going to see that we don't need neural stimulation to get cardiac contraction. It has a built-in pacemaker activity, okay? When you do heart transplants, drop a heart into somebody else, it's now devoid of neural connections. It still has a rhythm, okay? Skeletal muscle, it requires neural stimulation to tell it when to contract, how long to contract, and how strongly to contract. It doesn't do that on its own. With cardiac muscle and smooth muscle, you're going to see that most, but not all, has an intrinsic contractile activity. They are modulated by hormones. They're modulated by the autonomic nervous system, but this is modulated purely by what's called the somatic nervous system, the somatic motor nervous system. So, let's look at this, and so we have depolarization of this axon coming down here to the synaptic terminal. So those action potentials going across the membrane of the synaptic terminal will open those voltage-gated calcium channels that sets off that whole process, causing the exocytosis of those vesicles that are filled with acetylcholine. So that acetylcholine will diffuse across the synaptic cleft, and it's about to bind with these ligand-gated nicotinic acetylcholine receptors. So once that acetylcholine binds, then we have sodium flowing in. That's going to lead to an end plate potential, If that depolarizes the membrane enough that that current can spread down the T-tubule or along the sarcolemma, out here is where you have voltage-gated, and down here, voltage-gated sodium and potassium channels, and therefore you can have action potentials. So, here's that ligand-gated acetylcholine. You've seen this picture now many, many times. Seen this many times. So acetylcholine binds the ligand that opens up that channel. So, you open up that channel, we're generating an end plate potential, and then action potentials can, and I didn't draw in the action potentials, I'm just showing the wave of depolarization, can sweep down the T-tubules along the sarcolemma to activate another T-tubule over here, and that goes pretty fast. So here we're going to have that activation, that depolarization is going to activate that dihydropyridine receptor, which activates the ryanodine receptor, which opens up, which I threw down in here, which opens up a calcium channel to allow calcium to flow out of the sarcoplasmic reticulum by simple diffusion. So that's what's happening here. We have calcium streaming out of the sarcoplasmic reticulum. And once it gets into these sarcomeres, and it binds troponin, it shifts tropomyosin, you have thin and thick filament engagement and the power stroke, then those sarcomeres shorten. So once you get enough calcium, don't worry about the concentration. You need to have a certain amount of calcium to really kick this off. So there's a whole number of steps here. And if you have a problem in any one of these steps, and you'll be surprised from up here to down here, how many different disease, and to here, how many different disease processes are involved in this. So, how do you get all that calcium from the sarcomeres back where it belongs? Because as long as there's calcium in there, that muscle's going to keep those cross-bridge heads. As long as there's calcium and as long as there's ATP, those cross-bridge heads are going to continue to cycle, and you're going to continue to generate tension. How do you get that muscle to relax? Well, you have primary active calcium ATP pumps, ATPase pumps, these are working 24/7, 365, even during the contraction. These things are pumping calcium back in here. It's just that when you have the opening of those those calcium channels in the sarcoplasmic reticulum, they release so much so fast that what's coming out, what's effluxing, is coming out faster than what is being transported back in. So during this period, is that calcium ATPase pump working? Yeah, it is. But the release of calcium is so much greater, the efflux of calcium is so much greater than the influx, that you get this buildup of calcium and you get the muscle contraction. So it doesn't take very long, it's just a few milliseconds. Once the action potentials stop, you close those calcium channels on the surface sarcoplasmic reticulum. Boom, just like that. So that efflux stops, and then therefore, very quickly actually, you pump in enough of this calcium to get above that 10 to the -6 molar calcium concentration, and then you get muscle relaxation. Okay. So, now, once these action potentials stop, that acetylcholine just doesn't continue to work forever. I mentioned it briefly. The other day, and I didn't mention it earlier when we were reviewing that slide. There is an enzyme that's being released by the skeletal muscle at the motor implant called acetylcholinesterase. So it will degrade that acetylcholine. Once a neurotransmitter does its job, you don't want it sticking around forever. So here in this synaptic cleft, that's how this is dealt with, by acetylcholinesterase. It breaks it down very, very quickly. So that will stop the graded potentials, in-plate potentials, stop the action potentials. So it's going to look something like this. In-plate potentials leading to action potentials, leading to signal to release calcium, and then it binds troponin, tropomyosin shifts, we get engagement and power stroke. So as long as that neuron's firing, you can have episodic contractions, or you can have one continuous contraction. You know, if the nerve is firing intermittently, you can do this. If the nerve is firing continuously, you can have that type of contraction. Okay, so Dana came in and she was talking about reading a book about myasthenia gravis. So very quickly, I decided to put together a whole bunch of slides to talk about. They were already here. Well, I guess they're in your handout, so you know. So myasthenia gravis is just one example of where you can have a pathological condition associated with this system. And you're going to actually learn about quite a few in clinical pathophys. But you know, as long as you understand these basic mechanisms, you'll definitely understand, oh, we have a problem here, it's going to cause a problem with everything downstream. So myasthenia gravis, if you look at the name, it's IA means condition of, stand means strength, A means no. Asthenia means no strength. This is my for muscle condition, no muscle strength. Okay. And so what happens is there's this autoimmune destruction for whatever reason, don't know what the trigger is. At least I don't think it's finally understood. Probably a number of different things. But there's an autoimmune destruction of those acetylcholine nicotinic receptors that are found at the motor implant right here. So here's your body's own immune cells that, for whatever reason, they recognize this motor implant and proteins there more, and what's really being destroyed the most are these ligand-gated receptor proteins, these nicotinic acetylcholine receptor proteins. For whatever reason, they're goes in and what does it do? It starts degrading those nicotinic receptors. So, do you think your ability to signal from the neuron to the muscle is as good as what it was before? No, it's not. It goes down, goes down, goes down. So muscle weakness is very commonly seen being uncoordinated, because doing these movements, just this movement I'm doing right here is incredibly complex. Things going on at the shoulder with the flexors and extensors, at the arm flexors and extensors, at the forearm. That's a very complicated movement. And these things don't tend to happen uniformly everywhere at once. So if one area has an attack going on and you're losing more of these at a time than, say, your antagonistic muscle, you can see that it would lead to some discoordination. But primarily, there's a loss of strength. There's a loss of strength. And, that's some pretty interesting stories that I'd love to tell you about people, how they first noticed. Oftentimes, not always, but oftentimes, it might be muscles in your eye muscles that control the movement of your eye. And, you're going along, and all of a sudden you have double vision. And you go, man, what's going on? You look in the mirror, and one eye's starting to go one way, and the other eye's still fine. Gee whiz, what's happening? And it's not unusual for sometimes the first symptoms of myasthenia gravis to be in the face and eye muscles. Not exclusively, for sure, but we use those things all the time. So if something starts happening and your vision changes, and you go, man, all of a sudden I'm looking at two different things with my two different eyes. They're not coordinated anymore. Well, it's not unusual if that's happening. So, what's one therapy for this? Well, since you have less nicotinic acetylcholinergic receptors, one thing you can do is just have the acetylcholine stick around longer. So you use the receptors that are still left longer. So one of the first line therapies for years has been to give an acetylcholinesterase inhibitor. So this is just one of those things that if you understand the basic function, yeah, therapy kind of makes sense. Therapy kind of makes sense. Okay, cardiac muscle. Cardiac muscle, there's a lot of things similar with skeletal muscle, but it also shares characteristics with smooth muscle. So skeletal muscle, it's multinucleated. The muscle cells themselves are, but with skeletal muscle, it just has one nucleus for each muscle cell. And it's striated. You have thick filaments and thin filaments. The cross-bridge interaction is very similar. Cross-bridge interaction is very, very similar to what you have with skeletal muscle. You have troponin, you have tropomyosin. One thing different is one cardiac muscle cell is functionally linked to another cardiac muscle cell by these intercalated discs. They are fused together. And within these discs, there are these desmosomes, that's the firm attachment, but also within the desmosomes gives you the firm attachment, but also within the structure, you have these gap junctions. These open areas that allow communication from one cell to another. And what can flow through there very readily is electrical current. So if this muscle fiber, cardiac muscle fiber, is activated, it's depolarized. That depolarization can pass down to the adjoining muscle. So they are electrically connected. That's not really the case with skeletal muscle. We don't have these areas of low electrical resistance. Cardiac muscle is just jam-packed with mitochondria. Not surprising. You need a lot of oxidative energy production in cardiac muscle. So much. But here's a picture that I want you to take a figure I want you to take quite a bit of time studying. So here we have cardiac muscle, and over here, this cross-bridge cycling, force generation, sliding of thin and thick filaments, this over here from 5 and 6, this is very much the same as what we just saw with skeletal muscle. So from the time that the calcium engages troponin to tension generation, you already know this. You already know this. So, how is cardiac muscle activated? What's similar, what's different? So, whenever we have a depolarization of the membrane, let's say an adjacent cell sends an electrical current, and what does it do? You have voltage-gated sodium and potassium channels up here on the sarcolemma, of the plasma membrane of that cardiac cell. So if you have a current coming in that will depolarize that voltage-gated sodium channel to threshold, guess what? You get an action potential. You recharge the charge of that membrane. So when you have an action potential here, that current can spread, and you have these voltage-gated calcium channels here on the surface. And these are called L-type calcium channels. They also mean slow type. But when you get an action potential, it's going to depolarize the membrane here and cause that voltage-gated calcium channel to spring open. Now, one thing different between, and like I say, just keep going back to that table. One thing different between skeletal muscle and cardiac muscle. Cardiac muscle needs some extracellular calcium to get everything going. You didn't see extracellular calcium with skeletal muscle. It was sarcoplasmic reticulum calcium. With cardiac muscle, you need both extracellular calcium and you need sarcoplasmic reticulum calcium. Okay? So, when we have a membrane depolarization going on, that's going to pop open this L-type calcium channel. It's going to let some extracellular calcium come in, and it's going to bind a ryanodine receptor on another type of calcium channel. This is very much like the calcium channel you saw from the sarcoplasmic reticulum in skeletal muscle. But here, it's calcium binding, guess what? A calcium receptor. Extracellular calcium is binding this sarcoplasmic reticulum calcium receptor, and what happens? It pops open, and then huge amounts of calcium can come flooding out into the cardiac muscle cell, and then 5 and 6 go on. So, depolarization of the membrane, yep, that's similar to skeletal muscle. Opening of a membrane, voltage-gated calcium channel, and you do need to know that term, L-type calcium channel. That's going to spring open. Extracellular calcium is then going to signal the calcium channels in the sarcoplasmic reticulum to open up and let calcium out, and then it's everything you've known before as far as cross-bridge cycling with skeletal muscle. Yes?
What causes the first depolarization?
There would be another, imagine another cell. We have those intercalated discs. So we had a cell over here that had action potentials that passed here, and that current can pass, and it drives that to threshold, boom, then you recapitulate, you start it in this cell. So remember that it all starts, you have pacemaker activity, remember that? You have areas of the heart that will spontaneously depolarize, and that polarizing current will spread. It'll spread down a conduction system. It'll also then, once it gets within the muscle, spread from muscle cell to muscle cell. So when this muscle cell depolarizes and has an action potential and starting its contraction, that current has spread to the next muscle cell already. Current spreads quickly. And that muscle cell is now generating an action potential and recapitulating. So there'd be another cell here and another cell here, and they can spread via those gap junctions at the intercalated disc. That electrical current can spread. It's somewhat analogous to, you know, when you're going down a myelinated axon, that current spreads to node, action potentials, node, action potentials. Well, here it's actually going from cell to cell, but you get an action potential here in this one cell, it spreads across to the next cell. You have another set of voltage-gated sodium potassium channels, boom, an action potential there to charge that membrane, which will then send that current to the next cell. And it happens very, very rapidly, and you know, you have between your atria and your ventricles, it's just a wonderful system using the conduction system, and then finally the muscle-to-muscle communication system. Now, you have a whole, obviously, when your ventricles contract, it is absolutely vital for them to then rapidly relax. You know, sometimes relaxation is just as important as contraction. And with the heart, you want to have contraction and then relaxation. Contraction and relaxation, because it's during that relaxation period of your ventricles, that's when those ventricles fill with blood. If they don't have time, if they don't relax sufficiently to fill with blood, then during the contraction phase, there'll be less blood to pump out into the vasculature, right? So you need to have nice rapid relaxation. You have this in your book, it just shows us very simply, but you have these calcium ATPase exchange pumps that will pump cytosolic calcium out here into the extracellular fluid, and it will also pump calcium back into sarcoplasmic reticulum. Last time I checked, and I have to be honest, it's been a few years since I checked, there are something like 7 different types of pumps and transporters that are active in getting calcium back to where it belongs. So your book just shows these 2, and they're calling them the same. That's perfectly fine. But these guys are working 24/7, 365. It's just that when you have action potentials here, opening up this gate, causing calcium, a calcium-induced opening of the calcium channel, this calcium effluxing out just overwhelms the action of these. Once this depolarization stops, that channel stops, calcium drops to a critical level, and that channel stops, and then these pumps, during diastole, allow calcium to go back where it belongs. So there's a lot of similarities, but there are some very important differences between skeletal muscle and cardiac muscle. We're going to see that more calcium leads to more forceful contractions, increased contractility. And we're going to see that sympathetic stimulation is going to impact what's going on in this slide. So you get more calcium in, and actually it's a more vigorous contraction and a more rapid relaxation in everything you're working with. Now, I already showed you this with skeletal muscle, how rapidly you have depolarization of the membrane and then tension generation. It looks a little bit different. The action potential and the tension generation curve looks a little bit different with cardiac muscle. Here is the green's the action potential, and why is that elevated so long? This has to do with calcium. This extended period when it's depolarized is because of that extracellular calcium coming in. That's leading to this long period before you have rapid repolarization. And yep, there's a short latency period, like we saw up here, before you start seeing tension generation. But because of those L-type channels allowing calcium to come in, the action potential and the contraction phase is almost exactly the same. They're almost exactly the same. They look quite different with skeletal muscle, but with cardiac muscle, they look very much the same. Very much the same. And that's all a good thing, because you can't fuse cardiac muscle contractions, because we're in an absolute refractory period all the way over here before we get another one, and therefore you can't fuse contractile forces like you can with skeletal muscle, which we're not going to talk much about. Smooth muscle. Smooth muscle, there's such a... Now, I'm thinking all of you probably read this on smooth muscle. How many of you read that in smooth muscle and go, I don't know if I know much about smooth muscle? Smooth muscle is quite varied. There's different types of smooth muscle. Yes, there's multi-unit and signal unit smooth muscle, but smooth muscle and some smooth muscle, depolarization of the membrane will cause contraction. Other smooth muscle, it doesn't. It relies on, for example, ligands binding to receptors, causing an IP3 calcium signal transduction process. There's quite a bit of heterogeneity with smooth muscle, but there are some things that you can talk about, that things are in common that you can kind of hang your hat on. Now, smooth muscle is unlike from skeletal and cardiac in that it's not striated. It does have thin filaments, it does have thick filaments. That's similar, but they're not arranged to look like striated muscle. And cardiac muscle and skeletal muscle are both striated. So you have these different dense bodies in smooth muscle, and these are kind of anchor points. Anchor points. You don't have sarcomeres here. You don't have, it's just arranged differently, that you do have these anchor points where the filaments will anchor to. And when smooth muscle contracts, because of these different anchor points, most smooth muscle isn't a simple shortening. Oftentimes, it can be, it depends on the geometry of where you find the smooth muscle. Sometimes it's shortening, sometimes it actually is, with smooth muscle contraction, the muscle goes out. Sometimes it's showing right here, it flattens out. But it is contraction, there is tension generation. There is contraction, there is tension generation. So you do have cross-bridge cycling. Just, you know, cross-bridge cycling, but the players for cross-bridge cycling, they're different. How do you signal cross-bridges to cycle and smooth muscle? It's different than in skeletal muscle and cardiac muscle. Because in skeletal and cardiac muscle, the calcium binding protein is what? Somebody, anybody? Troponin. Okay. Here, it's calmodulin. And here, it's quite different with how the cross-bridge heads will ratchet. So, this is what I put together, and your book has an all-encompassing slide. And this is the way I always try to tell people, how does smooth muscle contract, kind of one step at a time. So, something has caused an increase in cytosolic calcium. Okay, that's similar to what we've seen before with skeletal and cardiac muscle. We need calcium. But in this case, calcium is not going to bind this calcium calmodulin complex actually plugs into something called myosin light chain kinase. This is an enzyme. When you see kinase, you know a phosphoryl subunit's going someplace. So myosin light chain kinase, this is found on part of the head, not the tip of the head, but part of the myosin cross bridge. This plugs into that kinase and it activates it. This myosin light chain kinase is now activated. What does it do? Kinases will move a phosphate around. And what does it do? It phosphorylates that myosin cross bridge head. What does that do? That causes contraction. That causes contraction. So that's what's going on with smooth muscle. An increase in cytosolic calcium, binding with calmodulin, plugging into an activating myosin light chain kinase, which phosphorylates the myosin head, leading to muscle contraction. Now, I do want you to, and so it takes an ATP to do that. And I didn't draw that in. It takes ATP energy to do that. I do want you to take a look in your book at the latch, L-A-T-C-H, the latch phenomenon. The latch phenomenon is in that section of the book. You can have smooth muscle contraction that continues on for a long time and you think, boy, that'd be taking a lot of ATP. There is a way that some smooth muscle can remain in its locked, contracted position without constantly utilizing as much ATP. And that's with the latch phenomenon. So, smooth muscle, it tends to be a lot slower in contraction, but it can, the contraction in some smooth muscle can persist a long time. So, let's see if this kind of makes sense to you. Going back to what we talked about a long time ago with membrane signal transduction. You've already learned this. So let's say we are a resistant arteriole, so we're a smooth muscle that is found in an arteriole going to, let's say, your GI tract. Okay? So when your sympathetic nervous system's turned on and you're releasing norepinephrine, actually the blood flow to your GI tract goes down. Why? The blood vessel vasoconstricts. Why? Let's look at this. So, when your sympathetic nervous system's turned on, norepinephrine is being released from sympathetic neurons. And it'll bind adrenergic receptors. And we talked about that there's different adrenergic receptors. There's alpha-1 adrenergic receptors, beta-1, beta-2, beta-3 adrenergic receptors. Here we have an alpha-1 adrenergic receptor, and if you remember, it is linked to a GQ, G protein. It will, when activated, remember the alpha subunit with the GTP comes over and binds the catalytic enzyme. In this case, it's phospholipase C. And I did draw in diacylglycerol, but you're also generating diacylglycerol. But remember, this pathway generates IP3, which signals the release of calcium. Now, you don't have an elaborate sarcoplasmic reticulum inside smooth muscle, but you do have other organelles with it, like endoplasmic reticulum. So that IP3 in smooth muscle is signaling, is binding different receptors inside smooth muscle, causing the release of calcium in the cytosol. And then that goes through what we just talked about. So, that is why, with those smooth muscle beds that have alpha-1 adrenergic receptors, you get smooth muscle contraction. Smooth muscle contraction. Now, I kind of want to, this is kind of the yin, let's kind of look at the yang about it. There are some smooth muscles that don't have alpha-1 adrenergic receptors. They have beta-2 adrenergic receptors. And either epinephrine or norepinephrine, either of those will bind these. There is some affinity differences. But when you bind kind of beta-2 adrenergic receptor that's found on smooth muscle, you don't get smooth muscle contraction, you actually get smooth muscle relaxation. Think about this. The same molecule, norepinephrine here, causing smooth muscle contraction, the same signaling molecule, same first messenger molecule here, causing smooth muscle relaxation. Why is it different? Different membrane receptors have different signal transduction pathways. That's what we talked about early on. So with beta-2s, the norepinephrine binds a beta-2, it activates a GSG protein that it activates adenylate cyclase that leads to cyclic AMP generation. The generation of protein kinase A will phosphorylate myosin light chain kinase, and what does it do? It decreases myosin's light chain kinase to calcium calmodulin. And I mean, there's just no affinity to it. And because it no longer wants to bind it, this myosin light chain kinase stays inactivated. If it's inactivated, there's no phosphorylation of myosin, no muscle contraction. You have relaxation. So, this kind of leads to the next section, which we're going to talk about, is the autonomic nervous system. Here, the autonomic nervous system, with the same signal, an increase in norepinephrine, or you could even put in an increase in epinephrine, in different smooth muscle beds throughout the body, you have quite different functional results. You have smooth muscle contraction here, you have smooth muscle relaxing here. Yes?
So is vasodilation just relaxation?
Vasodilation is a lack of, well, yes, vasodilation is relaxation. Yes, it is. Yes, it is. I was going to say it's a lack of contraction, but it's actually because they're usually tone. So basal tone. If you keep this turned on long enough, that basal tone even relaxes a little bit more. So some blood vessels, you're going to see strong vasoconstriction responses to some things, and they have vasodilation responses to others. So with smooth muscle, this is the way they show it, and it's pretty much the very same thing as what I showed you. Cytosolic calcium, activated calmodulin, it activates myosin light chain kinase, and that will lead to cross-bridge interaction and smooth muscle contraction. And they do throw in here that there is, how do you remove the phosphate, how do you turn things off? There is a myosin light chain phosphatase, which will reverse the process. And we might actually talk about that later on. Here is a figure in the book that it's starting to look at some of the similarities, some of the differences between smooth muscle and cardiac muscle. Cardiac muscle, or excuse me, skeletal muscle and smooth muscle. You're going to see that cardiac muscle and smooth muscle, many of them have rhythmicity. They have pacemaker activity. Not all of them do, but I do want you to take a look at in your book about some smooth muscles have the ability of generating their own action potentials and self-activation. Now, not all smooth muscle, but some smooth muscle have gap junction, so that stimulation of one smooth muscle fiber can activate the adjacent smooth muscle fiber. But that's not the case with all. What this is showing is really innervation of smooth muscle. There are all these variosities that basically you have the release of neurotransmitter in an area, and there are adrenergic receptors on here. It's so hard to categorize very cleanly smooth muscle. It's just so difficult. I do want you to please read differences between single unit smooth muscle and multi-unit smooth muscle, because that's going to pop up a lot. I want you to read that and be comfortable with it. This is where we started with this section, and I hope that with the textbook and what we talked about, you'll spend some time, quite a bit of time, looking at what's similar and what's different, because what's similar and what's different here will make understanding certain muscle pathologies much easier to understand, and also different drug therapies, okay? Now, for just a couple minutes, I want to just introduce the idea of the autonomic nervous system and go back to this, what we saw before. The autonomic nervous system is actually part of the efferent division of the peripheral nervous system. And the autonomic nervous system, this is a diagram that I hope you practice in shorthand, jotting this down in 10 seconds or less. There's so much information that can help organize your thinking, and the more you practice writing this down, the better. I hope you practice writing this out in a shorthand style too. I have a lot of students that have figured out ways of doing their own shorthand, doing this in 15 seconds. You don't want to put a whole bunch of stuff here, but start now with, okay, there's some similarities and differences here. So this might be as much as what a person writes down. So we're not going to get very far in this, but here is looking at the efferent division of the peripheral nervous system. You have the somatic motor nervous system, that innervates skeletal muscle. And then you have the autonomic nervous system, comprised of the parasympathetic and sympathetic nervous system. This innervates muscles, glands. It can be heart muscle, smooth muscle, glands. And so up here, we've already talked about this guy. Your somatic nervous system, this is innervation of skeletal muscle. Innervation of skeletal muscle. So what you see here, and we'll pick this up again, is from your spinal cord, so this is going to come out, this is going to come out via the anterior motor nerve root. It's a single neuron going to skeletal muscle. Single neuron going to skeletal muscle. It could be an A alpha nerve fiber. It could be an A gamma nerve fiber. So there's alpha motor neurons, there's gamma motor neurons. You know, the alpha motor neurons are going to innervate extrafusal muscle fibers. Gamma motor neurons are going to innervate intrafusal muscle fibers. In both cases, the neurotransmitter is acetylcholine. That acetylcholine is going to bind those nicotinic receptors we saw on those slides for skeletal muscle. So this right here would be the motor end plate. And the action of somatic nervous system activation is always excitation of skeletal muscle. Skeletal muscle, there is no relaxation response with neural stimulation. If you activate the somatic nervous system, you're going to excite skeletal muscle. We're going to see down here, well, you already saw an example with beta adrenergic stimulation. You get relaxation or inhibition of smooth muscle contraction. So down here with autonomic, sometimes it's activation, sometimes it's inactivation. But with the somatic nervous system, it's a one neuron going out to its target organ. Contrast that here with, well, these middle two. Let's just first look at these middle two. For the parasympathetic and sympathetic, it's a two-neuron chain. It's a two-neuron chain. With the parasympathetics, you have a long first neuron and a short second neuron. With sympathetics, you have a short first neuron and a long second neuron. And right here we have synapse points between this would be, well, let's just kind of, I'm going to back up and mention some things here. So whenever you have a neuron that releases acetylcholine, that's said to be a cholinergic neuron. A cholinergic neuron. cases, it's acetylcholine. I already mentioned that right here's that motor end plate. This is where you're gonna have those nicotinic, that's what the N stands for, nicotinic receptors. Those are at ligand receptor protein.